In this lesson, we'll talk a bit about how you can analyze the epigenome. We'll focus on a technique called ChIP-seq. Except where otherwise indicated, this content is available for use under CC BY 40. In this lesson, we will discuss the motivation of chromatin immunoprecipitation sequencing, known as ChIP-seq, which is an experimental technique used to study protein interactions made across the genome. This methodology has become a powerful tool in how we study the epigenome. Before exploring the motivation and steps to ChIP-seq, we will discuss the critical role the epigenome plays in gene expression. Health outcomes and who we are as individuals is not solely dictated by the DNA we inherit from our parents. Epigenetics has shown us that the choices we make directly and indirectly represent exposures that influence the epigenome to alter gene expression. These exposures are broadly classified as the exposome. The exposome can be a result of our ecosystem, social and socio-economic status, as well as physical-chemical interactions. These external environmental interactions eventually influence our internal environment, which dictate our life course and can impact our health outcomes. In this lesson, we will take a deeper dive to how we study the internal environmental changes through a method known as chromatin immunoprecipitation with sequencing, or ChIP-seq. This method allows us to study how protein nucleic acid interactions influence gene expression through the structure and function of the epigenome. To understand the sensitivity of the epigenome and our genomic landscape, we will discuss the importance of tissues we choose to study in ChIP-seq. We'll then discuss steps and principles in chromatin immunoprecipitation. Next, we'll discuss how sequencing the DNA attached to proteins through precipitation in ChIP enables the transition from ChIP to ChIP-seq. We will then see how researchers utilize the data to understand epigenomic and genomic regulation. We provide an overview of ChIP-seq by reviewing the steps and principles key to execution of the laboratory procedure. We will end with a forward look at how ChIP-seq has been advanced to further our understanding of the genome and epigenome. First, let's examine why tissue targets are important for studying the epigenome using ChIP-seq. Changes in our internal environment can be caused by our external environment across our lifetime. These changes often affect the balance between genomic and epigenomic regulation. The changes made to our internal environment affect the trillions of cells that make up the human body. These cells across the human body axis exist to carry out specialized activities key to life. This means that the external environment can cause disruption of these cellular activities and can have a disastrous effect on our health. Our understanding of cellular activities is linked to our ability to target the cells involved and their respective organelles. In many ways, a scientist's ability to target cell types and their respective organelles is analogous to a fisherman targeting an ideal location. They might choose a specific pond for fishing and the fishes of interest in that location. In general, it's very important to accurately select the tissues we plan to study, as they will influence the type of cells present. To understand the epigenome, we are interested in the internal changes occurring in the nucleus. Our understanding of nuclear activity will provide insights into how changes in our external environment and our lifetime exposure influences our health outcomes. The nucleus contains packaged DNA, and the internal changes that occur from our external environment could influence how DNA is accessed and subsequently transcribed. How the condensed DNA or chromatin is marked differs across different tissues and influences how genes are regulated across the human body axis. As we are studying the epigenome and its role in gene expression, we have to accurately identify which tissue we are interested in, because the chromatin marks can differ drastically. For example, a target gene in one tissue can be loosely packaged for easy access of transcription machinery for gene expression. In contrast, a different tissue could have the same gene packaged tightly, thus preventing gene expression. After identifying the tissues of interest to study, the cells that make up the tissue are isolated for further investigation. There are several experimental options one can use to study epigenomic changes, but for this lesson we will focus on chromatin immunoprecipitation techniques. Now, let's talk about a method that is important for understanding the epigenome. Chromatin immunoprecipitation, also known as ChIP, are experiments in which an antibody is used to collect nucleic acids bound to a protein. In other words, these are experiments in which DNA bound to a target protein is precipitated from the cell. ChIP experiments allow us to target the protein of interest using an antibody. Now, let's talk about the ChIP steps in more detail. Once we've isolated the cells we plan to study, we first cross-link the protein that is bound to DNA. This step keeps all protein-DNA interactions intact and prevents the proteins from separating from the bound DNA. Cross-linking steps are important in ChIP, as it preserves the protein-DNA interactions we intend to study. The cross-linking steps are completely reversible upon completion of the ChIP experiment. Next, cells are lysed and DNA sheared to allow for chromatin isolation. Proteins and their bound nucleic acids are inside the tissue cells. We need to break down the cell to allow for the proteins, including chromatin, to be released. Remember that chromatin is the densely packed mixture of DNA and protein. We could compare this to fish hatching from eggs. They are now available for us to catch. We will now add antibodies to precipitate our target proteins. The antibody we select should be able to identify the protein of interest. We are interested in changes made to chromatin that influence gene expression. Thus, we would want to use antibodies that recognize chromatin marks of interest in our tissue or other proteins that may influence gene expression. The antibody used in ChIP experiments is analogous to a fishing hook and bait used to target fish in a pond. Antibodies are added as they are able to bind to our target protein directly. For non-specific ChIP, antibodies that are not target-specific can be used. On binding of the antibody to our target protein, we use beads that are able to recognize the antibodies for purification. Bait and hooks used for fishing are analogous to our use of antibodies and beads. The fish is attracted to the bait, and when caught the hook is used to retrieve fishes bound to the bait. In other words, the antibody is like bait, and the beads serve as our hook to retrieve our target proteins. Next, we will clean and retrieve the chromatin. Beads facilitate the purification step because only antibodies bound to beads are selected. This step is similar to using a net or fishing hook to retrieve bound fish. At the end of the cleanup step, we are left with our target protein bound to DNA. We separate the DNA from the bound protein by reversing the cross links. We can use enzymes to break down the protein or the DNA, depending on which we'd like to study moving forward. We are interested in sequencing the DNA, so we will digest the protein. The isolated DNA fragments can be used to determine origin in the genome. This can help tease out the role of specific regions in epigenome and genomic regulation. Now, let's talk about the method known as ChIP-seq. Advancements in sequencing technology have allowed for high-throughput sequencing of DNA fragments isolated from ChIP experiments. This procedure is known as ChIP-seq. ChIP-seq is similar to ChIP, but there are added steps that allow for the preparation of DNA fragment libraries that are sequenced. We create these libraries by adding DNA adapters to the isolated fragment. We can then learn more about the region where the protein was bound in the genome. We can also learn if the DNA interactions are sequence-specific. How do researchers use ChIP-seq to make discoveries? Sequenced DNA reads from ChIP-seq are aligned to the reference genome first. This provides information about the DNA sequence location in the genome. Peaks can then be detected using algorithms like MAX. Motif analysis aims to identify overrepresented or enrich DNA sequence motifs within the identified peaks. These motifs are short, recurring sequences that are recognized by specific DNA-binding proteins. In epigenomic analysis, it provides insights into chromatin regulation. Differential binding analysis compares the identified peaks between different experimental conditions to identify regions where protein-DNA interactions exhibit significant changes. These different conditions could be a treatment versus a control or disease versus normal. Finally, researchers want to know if the peaks are close to known genes of interest. This helps them understand if epigenetic modifications are potentially turning these genes on or off. How has ChIP-seq played a part in better understanding the genome and epigenome in humans? The first human disease linked to epigenetics was cancer, with research showing that colorectal cancer tissues have reduced DNA methylation compared to normal tissues. This meant that many genes were turned on when they weren't supposed to be. Too much methylation can also be bad. For example, it can deactivate tumor suppressor genes that hold cancerous growth at bay. Fragile X syndrome is the most frequently inherited mental disability. It includes developmental delays, learning disabilities, and social and behavioral challenges, and it affects males more often than females. The syndrome is caused by methylation abnormality that turns off the FMR1 gene. Epigenetic treatments targeting reversible modifications like DNA methylation and histone acetylation offer potential in treating diseases. Examples of drugs include inhibitors of DNA methylation. Caution is needed because epigenetic processes and changes are widespread across the body. To avoid inadvertently inducing cancer in normal cells, treatments must selectively target irregular cells. Let's review what we have learned. In this lesson, we explored the importance of tissue targets when understanding the epigenome. We learned about the steps involved in ChIP and ChIP-seq methods. Finally, we learned how researchers analyze ChIP-seq data and how that data has helped researchers better understand the human epigenome with respect to disease and disease treatment. Thank you to our image and icon sources. Thank you to everyone who contributed.